Um, Excellent talks, everyone. Okay, the first question to Nick is from Bernard Blob. Are you working on turning the single cell transcriptomics data into a plant figure for in silico hybridization, or do you know of anyone who does?
It's a good question. It's a little bit tricky with single cell data because we're not sure where those cells exactly are coming from. You know, they're isolated from fact sorting. And the data, the T-SNE plot that I showed from John Schiefelbein's lab was very, it had some subclusters, and based on identity, I don't know if they used the ICI algorithm, whatever, they said, okay, these subclusters are these particular cell types. And we tried to, draw those back to graphics showing where on the roots those cell types are. But again, it's slightly problematic. I don't know if anyone has a good suggestion for how to visualize these things. I'm open to it and open to working with anyone to come up with a new visualization. Yeah.
Thank you. From Alexander Borowski, ePlant browsing is organized at a per gene level. How could we adapt the system to consider metabolites, processes, or other drivers of biological questions besides how does gene X behave for the PCA?
Right. So ePlant can display any sort of quantitative data and network kind of data, and we are quite close to actually rolling out an ePlant module for pathways in collaboration with Reactome, Plant Reactome, with Pankaj Jaiswal's group at OSU. So I think it's really, okay, if we have a framework, what can we put into that framework? What do we want to see? And it's really what scientists want to see, and anything is possible.
Cool, thanks. From Alexandria Tran, what are your biggest challenges working with multiple sources of data, and what are some of the guidelines you would like to see to make our data more scalable for databases like ePlant?
So certainly the curation is kind of the biggest challenge, I would say, in getting the data in some kind of format that we can, create a database. Sounds really boring, but it's really important. Having the proper metadata and the metadata not missing information, that's such a huge issue. There are other issues related to sort of sustainability of the of databases and that kind of thing that I'm not going to get into, but certainly like the curation aspect is huge.
Okay, great. Let's move on to questions for the next speaker, which will be coordinated by Jason. So this is for George.
Hello, my name is Jason Thomas. I'm a postdoc at the Relab at Carnegie, and I'll be asking questions for George. So the first question is from Sharon Greenblum, and she asks two things. One, are there meaningful statistics for describing and comparing multi-slice networks? And two, is this an active area of development?
Okay, great. Thanks for the question. Yes. The network science community, going back 7 or 8 years, has developed the theoretical framework for multi-slice networks. The most common application for them at the moment has been in transportation systems, where people look at the nodes being cities and the different slices being different modes of travel, be it road, rail, air, etc. And this has really come into its own in the understanding of the spread of the COVID-19 virus recently, in trying to inform policy and understanding how to control the movement of people, but its application to biological systems remains quite basic. I'm not sure it's actually been done outside of a yeast cell. However, the process should be directly translatable. And the second part of the question was, is this enacted? Yes, the network science community continues to work in this area. Doing things like community detection and clustering is actually non-trivial, but the mathematics are far ahead of where the applications.
Great. Next question comes from Amit Bakirbas, who asks, can 3D topological analyses help establishing identity of rare or unknown cell types for PCA if you don't know what the exact cell types present in that tissue?
Yeah, that's a great question. Going back to what was the question previously for Nick, which is the relationship between the organization of the cells and the data coming from single cell data and the capacity to merge these, so to understand where these cells are coming from within the tissue context. And to be honest, doing things like in situ hybridization with, say, rare expressed genes in these rare cell types might help to bridge that gap between the molecular and tissue scales. And I believe the group of Ryan Lister in Perth, Australia has begun to do this.
The next question comes from David Earhart, and he asks, what are the challenges in integrating multi-slice networks with physical data such as cellular mechanics or growth?
I would say probably many. I hadn't considered that. So, I mean, the extraction of cell geometries involve the use of meshes to describe surfaces. And from those, things like finite element modeling could be used and have been used in the past. But this is not something that has begun to be done yet.
Great, so with that, we will move on to our next set of questions. Thanks again, George, for answering that. So now I will be passing the mic to Emily, who will be coordinating questions for Amy.
Hi, everyone. Really awesome chats or talks today. My name is Emily Fryer. I'm in the Relab at Carnegie Institute for Science. The first question for Amy is from Alexandria Tran. As she asks, since one of your goals is very wide user engagement, what are the system requirements to run Yggdrasil? And are there aims to make a compact version that can be run on lower power computers when consistent web or compute access are not available?
Great. Well, thank you, Alexandria, for the question. So right now, the way that we have the framework set up is that we're doing as much behind the scenes as possible. Most of the compute is handled by our servers at the University of Illinois, our clusters, and we have access to the supercomputer here. So right now, all that users need to have is a desktop or a laptop computer. And that's something that we want to maintain so that, you know, something that we always have in mind is minimal burden on the user, and that includes computing power. The limitation there is that if folks want to run the models, they can download it and they can run it directly on their own system. And so that's variable depending on the size and the needs of that particular model. But yeah, we're just building an interface and having things run on ours. So one thing that we can think about in the future is making a mobile version that would be easier for folks to use just in their hands.
That's really cool. What are some of the biggest challenges for identifying your stakeholders and beneficiaries and reaching out to them to participate in this?
Yeah, so I think there are a lot of challenges, and the examples that I gave are the ways that we've been thinking about it, and it's not even all of the examples. We also consider trainees and how can we engage with broader communities, and so we've developed internship programs where we're taking a more STEM pathway approach rather than a pipeline approach. And so the way that we design the internship is that it can be done from home. So it works around the schedule of people that might be full-time students and full-time workers. And they can't get into the lab for 20 hours a week. And so it's a completely virtual, which that works out really well right now, a completely virtual internship so that it's more flexible. So that's something that we're thinking about. But what we've found to be the most useful is to have this free and accessible annual meeting. And what we have found, which was, I guess, a serendipitous thing that came out of it, is that it's really a lot of early career scientists that are joining this meeting. because it's low cost. You just got to get there. But it's a really great opportunity for early career folks to interact with those that are more senior, and then also engage with the tool and give us their feedback and their vision for needs in the future.
Fantastic. And then we have a couple more questions, but probably time for one. So I'm going to do a question from Ken. What about the critic stakeholders? Do you have specific ideas on how to engage them and give them a voice to the project?
Yeah, I do. It's something I think about a lot because we've been met with a lot of criticism. It was pretty difficult to get funding for our project because it has a little bit of everything. And so I think you have to get inside the head of the critic to find out why they're so skeptical of the work. Do they feel threatened, or that their contributions aren't being recognized? Are their needs not being met? And so the best way to do that is to directly engage with them. You've got to uncover the root of the skepticism, ask them for their advice and their feedback, ask them what we've overlooked, and how we can improve. And so I would highly recommend that for this effort, is to figure out who are your skeptics and what do they need and advise you to do. Bring them in.
Awesome.
Great.
That's really inspiring. Thanks, Amy.
With that, we will move on to questions for Drew, which will be coordinated by Kangmei.
Hi, everyone. First, thanks all the speakers for those great talks and insights. I'm Kangmei Zhao, a postdoc at Carnegie Institution. I will be asking questions for Drew. So we've got a couple questions. Actually, Lenore Reiser and Sue Ray have similar comments on the experiences of the successful examples like iGEM and SingBio. So since these two communities are very large and also mostly self-organizing, so could you please comment on the hardest part to establish the community, and also what do you think the most what is the power to nurturing the community?
Thanks, Kangmei and Sue and others for those questions. Let me take them backwards. So I think there were 5 things that helped the community grow organically. The one was working with the people who wanted to work together. We made the mistake initially of thinking, well, we have to work with these people because they're the famous metabolic engineers or genetic engineers or plant engineers or funding agencies. And that turned out to be a mistake because they're all too busy. Work with the people who wanted to work with us, who happened to be the teenagers who wanted to learn how to access the future of biotechnology. That was number 1. Number 2, we didn't tell them what to do. And this was templated off of Paulo Freire's experiences summarized in his book, Pedagogy of the Oppressed, working to bring mathematics education to landless peasants. Rather than defining what the projects were for the students, we said, well, tell us where you are and what's meaningful to you and do those projects. Okay, so that was a big deal. Third, giving and getting, a culture of sharing embedded within the technology core of the community. At the end of a project, the members of a team would contribute their best genetic gizmos to a collection that could then be shared freely with the next generation of teams. So that get and give was great and still is. Fourth, recognition mechanics. So when you think about the Olympics and the medal system at the Olympics, there's a gold medal and a silver medal and a bronze medal, and there's one of each. That's artificial. You could run a different Olympics where anybody who makes a personal record gets a bronze medal. Anybody who beats the Olympic record gets a silver medal. And there could be multiple bronze and silvers. Anybody who breaks the world record gets a gold medal. There could be 100 gold medals in the Olympics in one event. So it's just a construct. And in iGEM, we took the latter approach, setting benchmarks that anybody could clear if they worked hard, aligned with the community values and reinforcing those values, so that people could get recognized and bring that recognition back to their local communities. We got scaling of recognition. It was really wonderful. And then last, this is really important, if you try and grow a community, you can't make everything awesome. There will be problems. And if you're anticipating growth, you have to anticipate the next generation of problems and work on them in advance, because otherwise you'll be overwhelmed with problems. And I'm happy to discuss that more offline. Now, the other part of the question is, what was the hardest thing? And the hardest thing was creating the space, the place where people could see that it was okay to be, to do this new thing. And without that space, whether you were a professor or a student or even a critic, it wasn't clear that there was something real there that was worth your life energy. And so simply, creating etymological space, creating burrito-based lunches to bring people together, or a Zoom hangout now, gave everybody confidence that there was something happening here that was important.
Cool. The other question is also from Sue. So could you please comment on how can we better balance driving technology development while also remaining flashing curiosity driven science?
It's one of the biggest tensions I've seen as an engineer working in biology, because biology as a substrate tends to be overdriven by applications more than any other technology. It's often the technology of last resort, because the problems that get brought to biology's doorstep can't be solved with the hardware store or the electrical engineering department. And what that means is there's a sustained underinvestment in tool development. So it's a great question, Sue, and thank you, Kangmei, for representing it so well. Best I can say is, to the nerd audience, right? The improved tools lets us do more of the things we love, whether it's science or tinkering and engineering. And so it's just awesome to make better tools. However, when you interface that with the rest of the world that wants to know, well, what's this good for? How will this address hunger or climate or energy or medicine? It's very, very tricky to balance that. And you have to, in my limited experience, it goes back to creating that place, that critical mass community that says, tools are awesome. And if we're not leading with tools, we're just not going to be leading with what we're doing. And so we have to celebrate the tool builders and tool developers. I've never seen a situation where people as a scientific community overinvest in tooling. I've just never seen it. It's always underinvestment. So I'm not worried about overinvestment. I'm worried about underinvestment. That may be wrong in this case, but teach me how to have that problem. I'd love to see that problem elsewhere.
Cool. Thanks, Drew.